Today I'm here to present one of the tools I am developing during my thesis project, which is OliveAtlas. So why do we study the olive tree? Well, it is a crop of great socioeconomic importance in the Mediterranean basin, especially for the olive oil and the table olives production. In 2023, the production was of 20 million tons in that year worldwide, and from all that production, Spain was the top producer with 26%. Its cultive has 10 million hectares worldwide, and from that, 93% are located in the Mediterranean basin, and 24% of that are located specifically in Spain. So we know that olive oil is the most important source of fat in the Mediterranean countries, and it has also benefits for human health. So olives might have been exploited for 19,000 years, but its domestication occurred 7,000 years ago in the Middle East from the wild olive. There are more than 2,000 cultivars worldwide that are clonally propagated by cuttings or grafts, but from all of them, the most widely grown cultivar in Spain, and specifically in Andalusia, is the Picual cultivar. So right now there are 8 genomes sequenced. So we have Farga, which is a nice individual because it's a millenary tree, and this specimen of Farga has more than 1,000 years. Then we have also some cultivars that are actually planted for production of olive oil and table olives, like we have Picual, Arbequina, Lequino, Afrantoyo. There is also the genome of the wild olive tree. And then we have two cuspidatas that are very used in Asia because they are good rootstock. So despite all the 8 cultivars that are already sequenced, there are no many bioinformatics tools to assist breeding or investigation. So there was a web tool that was called ReproLive. It was the only platform for gene expression, despite the importance of transcriptomic data. It was based on Roche 454 sequencing, but is no longer accessible, so we cannot come with this one. We have all the data in OliveAtlas, so it is not lost, but we cannot access this web anymore. And right now, the main reference genomic portal for Picual is the Olive3DB. So that's the reason why we are going to base our study in Picual tree. But many olive genome expression studies that are published nowadays are based on the novel transcriptomes, so that is very difficult for us to be able to compare the expression among different experiments. That's why we developed OliveAtlas, which is a gene expression atlas for olive tree that is based right now on Picual genome. So we have several experiments included in OliveAtlas. We have 76 RNA-seq experiments that are organized in 11 different datasets. One of them are the organs of olive tree. Then we have also tissues. We have a data set with pollen samples at different stages of germination. We have a sample of mature seeds. We have also a data set that comprehends several abiotic and biotic stresses like cold, wounds, and verticillium in leaves and roots tissue. We have also that same data set, but split it in the 3 different stresses in case someone wants to study them further. We have also a big data set of 36 olive cultivars with different tolerance to verticillium. Another data set of response to drug in Suri cultivar, and this is the only one that is not using Picual genome. This was using wild olive tree because it was like that in the experiment, so we used the matrix as it was. And then the last one, we introduced it some months ago, and it's response to drug in Koroniki and Martina cultivars, and this one is also in Picual. So OliveAtlas is based on another tool that is called ECGDB. This tool was developed by Noé Fernandez, and it was presented in the last year's cycle of seminars. So if you want to know further about ECGDB, you can go to the YouTube channel and check it there. But I'm going to give you highlights because I think they are important for you to understand the full option of OliveAtlas, sorry. So it can be easily installed using Docker if you are using MacOS or Windows, or from scratch in a Linux computer. It is designed to minimize data upload and maintenance, so you don't have to have a big background in bioinformatics. It's written in PHP and do not require many dependencies. It provides a ready-to-deploy genomic portals. I'm going to show you a live presentation, and I'm going to prove to you that it's really easy to configure, like the basic configure. It includes a step-by-step documentation and example data that you can find out in the GitHub page. And it, independence of relational database, reduces the technical expertise and time invested. So I'm going to change. I hope my screen is showing. If not, please let me know. So here, okay, I'm going to move the toolbar. Okay, you are not seeing it, but it's blocking me. Okay, so here I have my local installation of ECGDB. And if I go to expression data, here I have the expression matrices that I am hosting right now. So this is my local web page. I can go to Gene Expression Viewer. And as you can see, I have these two files in here. So if I want to add a new matrix, all I need to do is just copy my data and paste it in this folder. So when I reload the page, they appear here, like you can see. And what do we need this file to be like? So it's a matrix, it's an expression matrix, just like this one. In the first column, we have the gene IDs, and then in the next column, we have the different samples and replicates of our experiments. So replicates must have the same name for the same sample because the CGDB can interpret it that way that they are together. Okay, and we use normalized data, so we prefer normalization with TPM, but that's up to you. So, okay, as you can see, it's very simple to load your matrix expression data, and ECGDB also allows the user to have multiple species or multiple versions if you want to. So all you need to do is create a folder. Like, for example, I'm going to create a folder for my Mango database. I'm going to move my Mango database here. And now I'm going to create another folder for my Olive one, moving again. And once I reload the page, I have Mango, and then I have Olive on the other side. So it will replicate the organization that you have on your expression data folder. So it's as simple as that. You don't need to configure any more farther than that. And as you can see, it's very user-friendly since it will put the first 5 gene IDs in this box. So if right now I have Picual Olive IDs, and when I change to Mango, I have Mango IDs. So that way the user can know which kind of ID he needs to put in the input. And then here, if you start typing your gene ID, it will suggest you which gene you are referring to. So you can just click on top of it and paste it to the gene ID's square. Or you can just copy and paste your list of gene of interest. It's okay both ways. So I'm going to show you this, this tool further. To that, I'm going to move to the actual OliveAtlas server, this one. So we go to Gene Expression Viewer, and here I have the 11 datasets that I talked to you about, but I'm going to focus on the first one. And I'm going to copy, I have here a list of IDs. Okay, so I'm going to paste them here. These are allergens, olive allergens. So I want to check in the picual tissues dataset if those allergens are only expressed in pollen tissue or if they are expressing in other olive tissues. So we go to get expression. And as you can see, the tool is very, very fast. So first of all, we have here a brief description. You can write here whatever you want to. I wrote what samples we are going to find in this data set. And then we have pictures of the different tissues or phenotypes or whatever you want to add, and links to the publication in case the person that consults them want to go to the original source. Then we have the first one of the visualization tools with each the lines plot. With this, we can compare the expression of multiple genes among all the samples of our dataset. Okay, so it's a very interactive tool, as you can see. This pink gene here, it's very expressed on pollen, both pollen samples, but its expression is so high that it's hiding the expression of the other genes. So we can think that maybe they are not expressing at all. So to prove that, we just have to put our mouse on top of the name, we click on it, and it will disappear, and all the plot will adjust to the new expression values. And now we can see that the orange and the red genes are very expressed at flowers and fruit tissues. They are not so expressed in pollen, they are also expressed in leaves, and that way we can analyze this better. And if we put the mouse on top of a point, we can see the value for all the genes in that sample, or our query genes in that sample. And we can also put our mouse on top of one ID, and if you see it, we can highlight that gene. We don't need to hide or show it, but we can also highlight it in case you want to keep the expression of the other genes, but you want to see this better. We have also introduced these tools header over here. You can zoom in, zoom out, and you can navigate with the plot. You can change the view to bars plot also if you prefer this view. And you can also download the plot in different formats. Then we have the expression cards. Here we can check the expression of each gene for all the samples of our dataset, along with pictures of the tissues in this case, or the phenotypes if we are working with stressors, or wherever you want to. And here, if we change to another gene, okay, this one is a nice example. As you can see, the flowers are the most expressed tissue in this for this gene. So it's in golden. The card will be colored in gold color. And now here, this gene is not very expressed in most of the tissues, so the lowest expression will be 0 and the card will be black. Then we have the heat map when we can compare the expression of multiple genes in all the samples of our dataset. And if we put our mouse on top of a category, we can highlight all the genes and all the samples within that category, within that value. Then we have the replicates plot, where we can check the dispersion of the replicates for each gene in each sample. So we can see if they are good replicates or if we need to be more careful with one replicate. So as you can see, they are pretty nice. And if you put your mouse on top of one of them, you will have the exact value of that point. And lastly, we have the average values table. So in this table, we have the mean value for every gene in each one of the samples of our dataset, along with the annotation. We can also add the annotation, in this case with Terten, SwissProt, and Interpro. But you can add all the databases that you want to. I'm going to show you that a little later. You can also link the gene ID to the gene page. So when I click on top of it, it will send me to the gene page. Here I can consult the genome browser. I can also see the functional description, and also I have access to the sequences of the proteins, transcript, and CDS. Okay, and you can also download the table in different formats in case you want to work with that in your personal computer. So I'm heading back to the presentation. I hope it changed again. And I'm going to continue in here because I want to show you another extra tool. We have developed this recently, and it's not available in OliveAtlas because we don't have a dataset that fits this, but it's a very useful and very graphical tool. So here you can consult the expression of each gene on your query gene list in the different tissues or cell types that you have in your dataset. So here we have an example of a skeletal tissue. This is from a mouse, if I'm not wrong. And here we can check the expression of that gene in all the tissues of the bone. And if we put our mouse on top of a name, it will be highlighted both in this scheme and in the general scheme too. So it's very useful to compare expression along tissues. So ECGDB is highly customizable. We need to change a couple of PHP files and a couple of JSON files to have everything working. Well, I mean, if you want to have the default settings, just place your expression data into the expression data folder and it will be working. But if you want to make any change or any customization, you can change just a PHP and a JSON file, and it will be That's simple, as simple as that. So here in this PHP file, which is the main PHP configuration file for the atlas, the expression atlas, we have the different tools. Here I am showing all of them in the default order, except for the description, because I didn't have a description for this sample data. And then if I hide the table by putting a 0 here, it will not show anymore. And if I interchange lines or cartoons by changing the number, it will change also. So it's as simple as that. You can customize it just as simple as that. And for further customization, you have a JSON file. This is for the expression atlas, as I said before. So once you open the JSON file, this one over here, all you need to do is put your expression dataset name in here. It has to be the exact name as it was in the expression data folder. This is the same dataset I showed you in the example, Picual tissues dataset. And this way, all the configuration that we make will be connected to that dataset. Then we can add a link to the gene page. Like I showed you before, when you click on top of the name of the gene, it will open the gene page. If you want to, you can add it. Or you can add a link to an external web in case you are using UniProt IDs or a Davidopsis or anything that is in an external portal. You can also add the dataset description. As I showed you before, all you need to do is write down a PHP file and put it in the corresponding folder. And in this PHP, you can write wherever you want to, and that will be showing in your expression gene result page. Then we can also add gene annotation in the average values table, like I showed you before. If you want the gene average table to be along with the annotation, you can add it with this variable here. And all you need to do is, again, put the annotation file into the corresponding folder. It's just as simple as placing files in the folder that it corresponds to, and then putting the name of that file into the JSON file. So I know it's a little chaotic right now, but everything is very well explained and detailed in the GitHub page. So if you have any kind of doubt, you can check it out, or even you can reach us out if you want to. Then we have the images, the expression cards images, where we need to, again, put our pictures into the corresponding folder. Here, there are my pictures. And then I need to link each picture to the samples we want to use for that picture. So here the name is roots. I'm putting roots and the name of the image is that. So now they are going to show together in the expression card visualization. We also have a JSON, which is a different JSON for the expression images, like the skeletal tissue I showed you before, because this one is a little more complicated. But so we decided to put it in another JSON file. I'm not going to go further into this, but I'm going to tell you how you can create the expression images, because it's not that hard. All you need is a different picture for each layer you have in your scheme. So in this case, I have 4 layers for a tomato slice. And all the pictures need to have transparent background, and then the tissue that is going to be colored with the expression needs to be in white, and the parts of the picture that are not going to be colored, like for example here the seeds or the peel or whatever we want to, are going to be black. So that way, when we configure the other JSON that I told you about, ECGDB will color each layer. Like here, the flesh has a blue color, and here it's more orange because the expression is different. So ECGDB is going to color each layer, and then it's going to put them one on top of the other like a sandwich, and it will automatically make the final picture. So here are some expression images example we have in our different portals. This is the skeletal one that I showed you before. This is an scheme for cherimoya, and we have also life cycles of different organisms. So Keeping with the JSON file, this is the last thing I'm going to talk about the JSON file. We have introduced recently the option for the user to change the color scale, because maybe you have multiomics data or single cell or whatever you want to add in here, not only transcriptomic data. So maybe you need to adjust the scale to your own data. So you can change them here in the JSON file. There are 3 lines, and you can change them, and that way you can change not only the colors and the order of the colors, but also the ranges within the colors. So it's very customizable. It will totally change your appearance. So ECGDB includes the gene expression atlas that I showed you, but also includes a lot of basic tools. And between them, we have the BLAST tool, which is also linked with the GeneSet sequence extraction tool. And we have the gene ID and annotation search, which is linked to the GeneSet annotation extraction. So right now, these tools are not available on OliveAtlas. They are available on Olive3DB because that was the genomic portal. But we want to make a change and put them in OliveAtlas. And besides that, we want also to add the other Olive that are available, the other genomes like Arbequina, Lequino, and the rest of them. So I am going to show you my local installation, okay? So this is what I'm developing right now. So here we go to BLAST. This is our input formulary. I'm going to copy the sequence I have prepared for this demonstration. Okay, so this sequence is a protein sequence for an allergen. It's the OliE6 allergen. And in the NCBI database where I extracted this sequence from, it says it's a PIQUAL sequence. But we are not so sure about that, so we are going to do a BLAST-P with all the proteins databases, including here the PIQUAL one, and just let it calculate. So as you can see, it's very, very fast also. So here are the selected databases that I chose. And here we have a graphic, a plot, a summary plot of our hits. So this is the PIQUAL one. This is the PIQUAL subject. So as we can see, it's not very similar to the sequence we have from the NCBI. We have a lack of here in the start. There is also a piece missing here at the end of the sequence. And then this piece is not very similar. It's 64% only. Then we have most similar, like Arbequina. This one is nice, it's shorter. Those are 100%, but they are shorter. And these 4 options of here are probably the best options. We have another Arbequina ID, a Cuspidata one, and Frantoio, and this is the Farga. So it's probably one of these, the actual sequence that we have from the NCBI, because we have 100% identity and all the sequences comprehend in our query sequence. Because here we are not gonna get more than 200 base pairs in this case, because my input sequence was a very short one, but in this case, the magenta color is nice, it's 100% of the length of the sequence. And then we have here a table, a summary table, sorry, where we have the description. All of them are annotated as OliE6, so that's nice. Then we have the species, we have the subject from, our target, our ID, and then we have some statistics. This tool works by placing the BLAST database into the folder. Okay, so only with that we will have this tool available also. And with this, we have also the sequence extraction. Both of them, both tools, works with the same database, which is the BLAST database. So here we can just paste the list of gene IDs that we want to check. I'm going to let the example, which is PIQUAL. So then I'm going to select the database we want the sequence to extract the sequence from, and I download it. I will just save it wherever. And once I open it, as you can see, we have here a FASTA file with the sequences we have extracted from our database. Okay, so other tool is the annotation extraction tool. Here, all you need to do is place your annotation file into the annotation file folder again. And that file is something like that, okay? You need your gene ID in the first column. In this case, those are Piqual gene IDs. And then we need to have the annotation with different databases in the following columns. So here I annotated it with Araport 11, and here we have the description for Araport 11, Swissprot, Tremble, whatever database you want to. This is a file you need to make by yourself. And with that, just by placing this txt file into the folder, I can check the information. So here I have some Piqual genes again. I'm going to check the Piqual database, and I'm going to search them. So here we have the table. And this is a table very similar to the average table without the values, but the annotation, and you can also download it in different formats. You can link it to the gene page again or to an external database like this case. You can also filter by column. You can select the rows you want to download in case you don't want to download all your results. And this tool is also linked with the search box in here. You can have the search box or you can have the search tool. Those are the same. And once you have your annotation file into the corresponding folder, this tool will also be available for you to use. So if we search for auxin, for example, it will find them, it will search for them in all the databases that we have. So since I have 7 annotation databases, it will find the auxin in all of them. And until we click on them, it will not search. So it's very fast because this is not, the results are not gonna search until we click on top of the name. So here I have again an annotation table. Okay, and that's what I wanted to show you about OliveAtlas. So just to finish, this is a work that I have done during several years, but it has to be continued. So now we are going to add a new Piqual genome that is going to be published in the next months. It's by a group in Jaén University, if I'm not wrong. So once they have it available, we are going to implement it in our database because Piqual was the only genome that was not organized in chromosomes, but this one it is. So it's gonna be a nice update. Then we are gonna also to add synteny between all the 7 cultivars. We are going to update OliveAtlas with the genomic references of the other olive trees, as well as genomic tools like BLAST, Genome Browser, annotation extraction, and all the tools I have shown you that right now are hosted in Olive3DB, and we are going to move them to OliveAtlas. We are also going to implement 60 RNA-seq samples from roots and leaves under salinity treatment, so new data sets. And lastly, we want to develop a tool to plot ideograms of olive chromosomes. So you can check for each one of these trees. You can check the chromosomes and where the genes, the subject IDs that you want to search, where they are located in the chromosome, so you can compare along different cultivars. So that will be all. Thank you very much for your attention, and if you can have any kind of doubt, I will be pleased to answer them.